Okay, so welcome back to the CompuCell3D workshop. Today we are going to learn about Boolean network modeling in CompuCell3D. Just a reminder, this workshop is being live streamed, recorded, and distributed on YouTube. Thanks to our supporting institutions, NSF, IUB, Complexity Institute, OpenVT, Glimprint, and previous funding from NIH. I am Pedro Cenci Dal Castel from Department of Intelligent Systems Engineering, and I am substituting TJ Sego. So this was his original presentation on MaBoSS integration in CompuCell3D, and I will be teaching that to you today. So let's start with what is a Boolean network model. If you don't care about the actual quantities of a species in a cell, for example, the level of a protein, how much a certain gene is being transcribed, etc., if you just care about yes or no questions, for example, the protein is present or is absent, the Boolean model is for you. So it is different from the antimony model, which is ODE systems, where you care about the level of a species. Here, you don't care about the level itself, you just care about whether the signal is present or absent. Some biological processes can be well described with a Boolean state, which means true or false, up or down, um, on or off, like gene expression and some signaling pathways. So there are just two states, the activated or true or on or up, and the inactivated state or false or down or off. Okay? So here we have nodes as objects. For example, in this beautiful model of cell cycle, our nodes are the cyclins, P27, and others. They interact via logics. For example, here cyclin B activates CDC20, CDH1 deactivates CDC20, so on and so forth. So these nodes interact via logics. If one node is up, the other node goes down, so on and so forth. And there are events, which are the transition of nodes between the two states they can be at. So from up to down or from down to up, these are the events that happen in this network. These changes are stochastic, and I will explain a bit how exactly that happens. All these behaviors and interactions can be inferred from experiments. So, for example, knockout experiments and treating a cell culture with a drug can inform you what are the relationships between these nodes. So, why is Boolean network appropriate for modeling, for example, gene regulatory networks? First, you can consider a gene transcription as either on or off. Of course, this is a simplification, but it's often a valid simplification. You can consider the presence of a transcription factor as either on or off. A gene might be on for a certain amount of time and then deactivates. So Boolean networks also incorporate time in a certain way. So for example, if a gene is on for a certain typical amount of time, you can incorporate that in your stochastic Boolean network. Um, it might take a certain amount of time to turn on, which you can set, and turning on or off is often probabilistic due to stochasticity in cellular processes. So for example, if you have only a few molecules of a certain transcription factor, there is a probability that they bind to the gene location, to a certain specific site in the gene to allow transcription. This is often probabilistic, which makes the stochastic Boolean networks appropriate. And for protein signaling, analogously, you can consider the signal either present or absent, and response of the cell to that signal being also true or false, binary. And also you have typical amounts of time that the cell takes to respond to the signal, and the signal lasts for a typical duration. Although everything is binary here inside the Boolean stochastic network, to a certain extent, you can still incorporate some those response dependence to your stochastic Boolean network. And we will see that in the exercise later. So here are a few examples of published models using Boolean stochastic networks. So this is probably the most robust biological application of Boolean stochastic networks using MaBoSS. Again, so MaBoSS is the tool we are going to learn how to implement in CompuCell3D today. And in this particular case, they study the cell response to DNA damage. So DNA damage takes place, and then the cell can change state to a proliferating state, can become drug resistant, can undergo ferroptosis, apoptosis, or become senescent. It contains 34 dynamical nodes, so it's a relatively big network. It includes key nodes like P53 and others that are promoter or suppressor of tumors. These models, and so what do they get with this model? So first, they replicate or they reproduce experimental results. And I think is um, this answers Clara's question from yesterday. So how do you validate these models? And especially a Boolean stochastic network, how do you validate something that is binary to experiment? So here they are measuring the percentage of cells that undergo apoptosis versus ferroptosis. So they do an ensemble average of the Boolean stochastic network, and then they compare the ratios of certain states with the actual measurements from experiments. So in the experiment, they will measure the percentage of cells undergoing apoptosis versus ferroptosis, and then they calibrate their model to achieve the same ratio. But what are the key discoveries that you can achieve with such a model? So here they identify a key node, central regulator of the network. So by simulating knockouts, you can detect what are the nodes that have most impact on the network behavior, or also by changing the rates up and down of the network, you can detect what are the nodes that have most impact on the behavior of the network. What are the nodes that have the most impact on the behavior of the network? They also help identifying positive feedback circuits, which are often potential targets for drug development. This particular model identifies XCT overexpression as a main cause of drug resistance. I am currently building a model in CompuCell3D that involves MaBoSS, and this is um, what is IAV again? It's influenza A virus. Okay. So influenza A virus infection in CompuCell3D, and this is the network that I am running there. So each cell contains this network and is solving this network. So every cell in the simulation. And what are they doing? They receive a signal like interference signal. The interference signal might make the cells resistant to the virus, and also they can get infected by the virus. They can detect the virus, which activates secretion of interferon, And the virus may contain proteins that disable all these immune machinery of the cell. So the actual network is much bigger than what you're seeing, but this is the overall like the main nodes of the network and what they're doing. And here I opted for using MaBoSS because the network is very big. It contains like about 30 nodes. So doing that in using Antimony would be very expensive because every single cell is running this big model. Also, most of these nodes have a binary impact. For example, only one viral genome is enough to trigger interferon response. And of course, there is a dose response to interferon, and I also incorporate that. So this is a partnership between our group and APL from Johns Hopkins, and we expect this to be published in 1 or 2 months. So yeah, let me do some cleanup here because I'm seeing complaints in the chat. Thank you. And there's also this paper here, Modeling Signaling Pathways in Biology with MaBoSS from one single cell to a dynamic population of heterogeneous interacting cells. So in this paper, they explore all the applications of MaBoSS currently. Of course, they do not talk about CompuCell and MaBoSS integration because that's more recent. But here they explore MaBoSS, which is the most basic, ensemble MaBoSS, and up MaBoSS, which allow you to do ensemble averages with your MaBoSS network. And also PhysiBoSS, which is an integration of PhysiCell and MaBoSS, which allows you to model agent-based systems where each agent incorporates a Boolean, a stochastic Boolean network. Now, let's introduce you what exactly is a Boolean network, and we'll start with the simple case, which is a deterministic Boolean network. So this is a uh, introductory example. So imagine we have three nodes, G1, G2, and G3. G1 activates G2, G3 activates G2, but only if both of them are active. So this is being represented by these two pointed arrows, and they say here that they interact like an AND operator. So you need both G1 and G3 active to activate G2. G1 deactivates G3. So this arrow can mean many things. In this case, it means that if G1 is false, if G1 is 0, then G3 becomes active in the future. And if G1 is 1, it's true, then G3 will become 0 in the future. Finally, if G2 is active, then G2 activates G1. So that's the basic structure of the simple deterministic Boolean network. So here we have a truth table, so all possible combinations of states of this network and what will be the resulting state in the next step of the network. So for example, imagine that all the nodes start with state 0. So G1 is 0, G2 is 0, and G3 is 0. So what is the next step? So G2 will not become active because both G1 and G3 are 0. G1 will not become active because G2 is 0. And G3 will become active because G1, its suppressor, is 0. So if we start with the state that is 0, 0, 0, the next state is 0, 0, 1. Is that clear? Any questions here? Clear. Okay. I'll go over one more example here. So let's suppose all the nodes start with state 1. So G1 is 1, G2 is 1, and G3 is 1. So because both G1 and G3 are 1, G2 will remain 1. Now, because G1 is 1, it deactivates G3. So G3 will go to 0. And because G2 is 1, G1 will remain 1 too. So if our current state is 1, 1, 1, our next state is 1, 1, 0, where 0 here is the state of G3. So this is this in the state transition graph. We have all the transitions happening from one state to the other, and we can see that we have a cycle. So if we start with 0, 1, 1, we get into a cycle, and the network keeps oscillating between two states. And if we start on any of these five states here, the network will deterministically fall into the 0, 0, 1 state here in the end. So you can reproduce oscillations using Boolean networks and also reproduce end fates. Like your cell falls into a certain state and it stays there forever. Is the rule that you need to read from G1 to G3 in that order? Good question from Clara. So you don't read them in order because you don't need to read them in order. The changes, if you're asking about the changes of the nodes, they all happen simultaneously. They are synchronous changes. But here, as they are represented in the table as G1, G3, G2, and G3, as you can see here in the top of the table. So yes, 0, 0, 1 is a steady state, as Savani asks. Yes, 0, 0, 1 is one possible steady state. One possible. There is another steady state, which is this oscillation between 0, 1, 0 and 1, 0, 1. What would happen if there was a disagreement between what a state should be? Yes, so if there is a disagreement, for example, I have two nodes and one node activates a third one and this other node deactivates this other one. So I think this is Clara's question. So let me draw this network to um, make this example here. I have node A and node B and here node C. Let's suppose A and B are both true, so they have value 1. And C is, I don't know, C is 1. No, C is 0, for example. And then A activates C while B inhibits C. Then what happens? You have to solve that in the rules. So for example, if you write your rule as being uh, C becomes positive if A and not B, then if you have B, C will never go positive. So it depends on how you write the logic. Yes, you're always using ands and ors. Yes. And nots too. You're also always using nots. So these are your basic operators. Is that how you encode strength of each activation and suppression? Yes. So you can, for example, in this case here, in this particular example, B is stronger than A. B is more powerful than A. Yes, this is one way of doing it. Of course, we are not going to use deterministic Boolean networks. We are going to use stochastic Boolean networks. And The difference here is that in stochastic Boolean networks, the nodes can switch state probabilistically. And the transition probabilities introduce randomness and control how fast the nodes change state. So time here is not as discrete, let's say. It's still discrete, of course, but uh, if I give one time step, it's not guaranteed that a node will change state. I may need to do multiple steps because there is only a probability of this node changing state. And also, if all the nodes have some residual probability of changing states, then this resulting graph is completely connected. You can switch from one state to all the other states in the network. Also, because you are using probabilities to change state, this also regulates how fast the nodes change state. So if you set a very high probability, these states will change faster. Yes, each transition has its own probability. You can specify each transition using MaBoSS. Yes. Okay. Next, why and when use stochastic Boolean networks? So here is as a MaBoSS user, these are the whys and whens from my perspective. So first, MaBoSS is extremely fast, which makes it a good alternative for network modeling when your networks are extremely big and don't require as much detail. Second point, often in literature, there is no information about the intensity of a molecule inside a cell and those response curves. So when you are using an ODE model to represent molecules, genes, transcription, etc., you care about the level and the intensity of that molecule. But then you go to the literature and you ask the literature, okay, how strong is the effect of this molecule, or what is the amount of this molecule? And often there is no information about it. And even if you have information about the quantity of a certain molecule, like an mRNA molecule, let's say you find a paper that measures that the concentration of certain mRNA molecule increases twofold inside a cell as a response to something else. Does that really do something? Like a twofold increase, is that enough to really impact the cell in a expressive way? Maybe should it be 100-fold? So how do you know? How do you know if a twofold increase in an mRNA molecule is enough to do anything? Let's suppose a protein increases by 1000-fold. Okay, we might assume that it does have an effect in the cell because it increased 1000-fold. But uh, by the time it got to a thousand-fold the default concentration, is it already saturated? Like, would it have the same effect if it was only 10 times the default concentration? Often we don't have answers for these questions. Um, Also, what is the dose-response relationship? So let's say a molecule increases by 10 times in number. What is the result effect of this increase? So does the cell respond in another variable by a twofold increase, by also a tenfold? How is this relationship? Is it linear? Is it saturating? And how do we know? How do we know how sensitive the cell is to that molecule change? So when these kind of information is absent, using stochastic Boolean networks is often a good idea. So in literature, you will mostly find a vague understanding of how things interact. For example, if we increase this thing, then this other thing increases or decreases. So how do you use stochastic Boolean networks? you look for the most important behaviors that you observe in the literature, and you incorporate that in your stochastic Boolean networks by saying if this thing is active, then this other thing will happen. And you can incorporate time in the sense that how long does it take once this thing here is active for this other thing to become active as well. So you can incorporate that, and this is often an information you have accessible in the literature. So for example, if I get time series in the literature about influenza infection on cells, I know that it takes like typically 4 hours for a cell to start expressing viral proteins after endocytosis of the virus. So this information is often available, and I can use this information to inform these parameters, these time parameters, to inform my stochastic Boolean network. Is everything clear so far? Is what I'm saying making sense? Okay, makes sense. All right, let's keep going. So a few warnings and caveats now. First, this is very new territory. So there are many things still that we're still discovering. And MaBoSS and CompuCell3D integration is also very recent in the history of CompuCell3D. Also, the content of this module is new, originally written by TJ, and I think this was only taught once. This is the maybe the second or third time someone teaches this, and I am also making changes and updating these slides. So TJ says, I am the only instructor who knows how to use this capability to my knowledge. So this is 1 year ago. Maybe now I am also one of these. Again, a link to the documentation is available on our CompuCell3D documentation. You will find all the information you need to incorporate MaBoSS to your simulation. And both TJ and I are not expert in Boolean dynamics. So if you want to get deep in the weeds of how stochastic Boolean networks work, then, I mean, we will help you, but we are definitely not experts. In my case, I see a hammer, I smash something. That this is what I'm doing here. I learn enough of Boolean stochastic networks to hammer something. I don't know exactly the specifications of the hammer I'm using. So what is the objective of this lecture? Is just cover enough material, enough background so that you can implement MaBoSS networks in your CompuCell3D simulation. Clara has a question. So before we move on to MaBoSS plus CC3D integration, I will read Clara's question and hopefully answer it. Using these only on-off methods also has an assumption. Instead of assuming saturating function, for example, it assumes that the concentration does not matter, and the smallest amount will have the same effect as a large amount. It is still an assumption and does not solve the problem of missing data, just a different approach, right? So I disagree that it assumes that the smallest amount will have the same effect as a large amount, because you actually don't care about the amount. You can interpret a low amount as zero. So no, it's not equivalent. It's not just a different approach. It is a completely different approach. It is assuming completely different things, yes, but it can interpret a small amount of a molecule as zero. That depends on how you design your network. Of course, it does not solve the problem of missing data, but it requires less assumptions of the system because when you build a model that has the levels of these molecules, then you have like twice the number of parameters, and then it's harder to, for example, do a parameter scan, and you have more moving parts to your system. So mathematically speaking, a MaBoSS network is more minimal than a ODE model, if that makes sense. All right, so MaBoSS and CC3D integration. Why do we do this? Because MaBoSS do not allow cells to move. It's simply a Boolean stochastic network. CompuCell3D provides all the capabilities of representing a cell explicitly in space, growing, dividing, and interacting with neighboring cells. So here we have an example of a simulation with delta notch signaling and growth and division taking place at the same time. Also, the interface between MaBoSS and CompuCell3D is very similar to the integration between Antimony and CompuCell3D. So all the commands are very similar. Of course, the syntax is different, but everything we have been doing so far in terms of incorporating antimony to our models, which is attaching an antimony model to a cell, modifying an antimony parameter, all this structure is basically the same. So building a MaBoSS simulation requires a network specification and a simulation configuration. So the network specification is where you set the nodes and the interactions between the nodes. So for example, in this network specification, we have a node cycling A or SYCA. We have another node like cycling B, and these nodes interact together via logics. So here we have not RB and not CDC20 and not open parentheses, something and something else and something or something else. So here we are using nots, ands, and ors to build the interaction between these nodes. And then there are rates. So rate up, which is which sets the typical time it takes for our node to go up if the condition is true. And if the condition is false, you'll have another rate up because you are setting stochasticity. Sometimes a gene may still transcribe something even if the transcription factor is not available, for example. So you have two rate up parameters, one if the condition is true and another one if the condition is false. Oftentimes, the else rate up we set to 0. And yeah, oftentimes one of these is 0 for simplification. We also have a rate down. So if the node is up, if the node is true and the condition is true, then you have one rate down. And if the condition is false, you have another rate down. So the basic structure of setting a rate is rate up is given a certain condition, a certain rate up, and if the condition is false, another rate up. And similarly for rate down. Anastasia is asking advanced questions, which I will cover in other slides. Derek is asking questions that I will cover in the next slides. And we also have a simulation configuration where we set the initial states of the nodes. Like a node might be set to true or false initially. And also we can specify certain parameters. So for example, we can have a certain rate as a parameter, and we also specify the value of this parameter in the simulation configuration. So for example, we can implement delta notch signaling using MaBoSS. So here, delta notch signaling involves contact between neighboring cells. If cells are sensing delta signaling from neighboring cells, they start expressing notch. And if they have notch inside them, notch inside the cells suppress expression of delta. This can also be linked to a cell cycle model. And by incorporating both the delta notch signaling and the cell cycle, you can model something more similar to, for example, embryo development. So our first exercise for today is open the delta notch simulation. So we built a delta notch simulation using Antimony last week, and we are going to use that for today's exercise. Because the structure is very similar to the antimony structure, we are just going to start with the simulation we already have and make changes on it to incorporate MaBoSS instead of antimony. So I will give you 2 minutes to download the simulation. And if you are having trouble finding the simulation, I will just put it in the chat again. So this is our original delta notch simulation implementing antimony, which is not Boolean stochastic network. The antimony is an ODE system. Okay, so everybody have the code, which means I will proceed. Now, keep your code open. So with split screen, take a look at the code, and here we will compare side by side what MaBoSS is doing and what you have in your code. So this is MaBoSS, and I believe, yeah, so this is the original delta notch simulation. So we have an antimony model. Um, Yeah, we have an antimony model, which are two reactions, rates, everything else. So there are many parameters here. Now in the left, I have the syntax for the Boolean stochastic network. And I also have my Boolean stochastic network here on the right side. And we are going to compare. So take a look at your antimony, and we will compare to the MaBoSS structure. So first, we need this import random, which we don't have in the antimony model. Then we need our network structure, which looks very different from the reactions we have in antimony. So in this network structure, we have our node, for example, node notch and node delta. What is going on in the node is delimited by these curly brackets. So, and this is very sensitive. So C code is very sensitive to brackets and columns, etc. So this is oftentimes you forget to close one of these, and the code will crash and won't give any errors because the model is within the string. So usually what I do if I want to add another node is I copy an existing node. So just to make sure I have all the symbols in place. Okay, so what we have here? We have node and then the name of the node, like notch and delta. And then inside the curly brackets, we have a logic. So in this case, we have our logic is two parameters, DAVG and D threshold. And because they are parameters, they go with a dollar sign before. So these are parameters. For referring to the logic, we use at symbol. So if our logic is true, then there is a certain up rate. If our logic is false, then there is another rate. And this is the basic structure of logic if something, else something else, with this question mark and this symbol that I forgot the name. Semicolon. Okay, thank you. Can you compile the code before plugging in your CC3D file? This is done automatically under the hood, Noshin. Anastasia asks, could you explain why we need a rate up and a rate down? Yes, so we need a rate up because we want the node to switch to up state, and we need a rate down so that the node switches to a down state. And both of them may depend on a certain logic. So if the logic is true or false, then you have two values for each. So you have in total four values. Based on the logic, if condition is true, the value will be 0, and if the condition is false, the value will be 1. Doesn't that take care of both cases? Yes, it takes care of both cases. But for example, I may want to introduce stochasticity, and if I am introducing stochasticity, these values might not be exactly 0, and they might be different between each other. So this is when you start to get into the weeds of how to actually model something and to get the stochasticity close to what is the observed biological behavior. So good question. Quite advanced questions. I see that you guys understand how it works. Could people ask ChatGPT to write the code to go in there? Yes, yes. And there is a software where you can draw the network, and then it converts the network you drew to a MaBoSS network structure like this. Usually I write them myself because I don't trust the output of these softwares, but that's definitely something you can do. Anastasia asks, could you also use different logic for rate up and rate down? Yes. So you can have logic 1, and then I'm just messing up with the code to demonstrate. So you can have logic 1 and logic 2 like this. I think. I could be I could be wrong though. I don't know. Let me run this and see if this runs. It should, but I'm afraid I could be talking nonsense, so I'm just going to test this. Okay. Oh yeah. No, no, this is sorry, give me a minute. Show project panel, MaBoSS, opening player. These are all good questions. That's why yes, it works. So yes, you can have two logics. You can have two logics. You cannot have two rate up and two rate down. This one I'm sure. You cannot have two different rate ups because it creates conflict in MaBoSS. And if it's not throwing an error, it's probably choosing one of them. And you don't know which one it is. So yeah, logics you can have different logics, but yeah. Rates you can only have one of each. What is that software called, Clara? Good question. And I don't know the answer. Good question. Maybe you guys can find that for me too. But in this paper here, what is the paper? This one. It covers everything that you can do with MaBoSS. So you'll likely find the answer there. Okay, having the structure done, now we move to the rates, which I already talked about. So let's review this. Basic structure is rate up is a certain value if condition is true and another value if condition is false. So the first value is the rate when condition is true, and the second value is the rate when condition is false. The condition again is given by the logic. Uh, you always have two rates. You have two upward rates and two downward rates, as I was answering in to Anastasia. So you may want to introduce, for example, just two slightly different rates because the effect of a certain signal is not very strong. Or you can set a small rate to represent stochasticity. So that's why you often need two rates. So in this case, for delta, when notch is present, we have a slow rate of delta when notch is present because notch disables delta expression. And we have a fast rate otherwise. So if notch is absent, then we have a very fast upward rate. Then we have two downward rates. So if notch is present, we have a fast downward rate because notch is suppressing delta, and a very slow downward rate otherwise because when notch is absent, delta keeps itself up. Yes, the rates can be anything between 0 and 1. And in fact, the rates can be anything between 0 and infinity because they are rates. They are not probabilities. They are probability per time. So you can set values above 1. Good question. Okay, now let's go to our configuration. So in Antimony, you have your parameters inside the model string. Here we have another configuration string where we set the initial values of parameters. So here parameters come with a dollar sign. So this is our parameter DAVG, our delta average that we measure from surrounding cells. We also have a delta threshold that we also have to define with these dollar sign. And we also refer to them with a dollar sign. So parameters, so things that are not nodes and they're not logics, they're not rates, rate up or down, these two here, they come with a dollar sign. So you can, for example, say notch rate equals 1. And then you can put this variable here, like this. So this is possible. This is acceptable. This is how you refer to variables. Okay. Now, the initial state of the nodes, you write as the name of the node, then a dot, and then I state. I for initial, so initial state equals 0. Uh, I think I didn't talk about how to refer to a node in the logic. It is simply by typing the name of the node with no symbol before. So nodes are plain words. Parameters have the dollar sign before, and logics have the at symbol before. Okay, so how to set up MaBoSS? So first, we need a random package because we need to set a random seed. You don't want your network always behaving the same way. So we initialize a random seed and we attach MaBoSS to a cell. So down here, I have my add MaBoSS to cell. And in this MaBoSS to cell, we are giving the model structure string, the configuration string, the time step, and the time tick. So what is time step and time tick? Time step is how many time steps you are doing every time you call your MaBoSS time step function. So in a given step of CompuCell, you step MaBoSS. And then how many steps MaBoSS do depend on this time step. So if you set time step to sorry, this is the time resolution of your MaBoSS step. So if you set this to like zero point 0.1, your MaBoSS network is more fine-grained, but it also takes longer to run. And the time tick is how often you evaluate the network. So if I set this to 1 equal to the time step, every step the MaBoSS network does, you read the values of all the nodes. By default, I set them to 1, both of them. Uh, but you may also, if your network is maybe too too noisy, you can try decreasing the time step. And finally, we are setting a random seed. So here is where we are using our random package to specify an initial seed for our MaBoSS model. MaBoSS supports deterministic and stochastic time integration. So the deterministic we already saw, every step, all the nodes simultaneously will change based on the interactions. And the stochastic integration, which is this one where you set probabilistic rates, is the Glapsy algorithm, which is the default option when you are using MaBoSS in CompuCell3D. Suvani asks if the time step changes the network from synchronous to asynchronous. No. If time step is 1, does it mean MaBoSS steps equals 1 Monte Carlo step? Yes, that's what it means. Yes, correct. Okay, so we attach our MaBoSS network to all cells of the simulation. So to do that, we use this command, self.add to cell. And here we specify the cell we are attaching the model to. We give the name of the model, which we use to refer back to this network model of each cell. Then we pass the string of the model structure. So our delta notch string here is the model structure, and we are also passing our config structure. Finally, time step, which is the resolution, the temporal resolution. Time tick, which is how often the network reads the value of the nodes, and the random seed. This is very similar to the antimony way. Yeah, so it should be easy to just change from antimony to MaBoSS. So to attach a MaBoSS model to all the cells, we have to do a loop over all the cells. So for cell in cell list, which I have over here. And then we do self.add and pass cell to it. So this is looping over all cells. So by the end of this loop, all the cells have a MaBoSS network attached. Now, how do we interact with the MaBoSS model? So we have the cells with their Boolean stochastic networks, but oftentimes we want to pass information to the network and get information from the network. And how do we do this? So we use several commands here, just like in the antimony case. So for example, to acquire the reference for a certain MaBoSS network, we use cell.maboss dot the name of the model, which in our case here is delta notch, but it could be any name you give to your model. And then we can get a node of this model by using these square brackets and giving the name of the node. So for example, to get node NICD, we call cell.maboss dot the name of the network, and then we pass, this is a dictionary, so here we pass a string, which is the name of the node. So here in our case, we will do cell.maboss.delta notch and then pass the name, for example, delta, and then we are getting the node delta. Again, this is not the value of the node, this is just the node. If we want to get the value of the node, then we do dot state. We do node dot state. To get a MaBoSS variable, we do cell.maboss dot the name of the model dot network dot symbol table, and then we give the name of the parameter. So for example, in our case, we have these parameters DAVG, which we want to pass to the network. So we will do this cell.maboss dot delta notch dot network dot symbol table DAVG here, which is the name of the variable, and then we do equal the average of the delta in the neighboring cells. So these commands you don't need to memorize. You can call them in here in your Twedit. So CC3D Python MaBoSS, and then you can go, for example, to get a MaBoSS network node, or so it gives this node equals cell.maboss.myModel. This is the name of the model, and then the name of the node. So to edit snippets give you everything. CC3D Python, MaBoSS, um, get model, get external variable here. So variable is a certain model, which is, for example, cell.maboss dot my model. This would come here, for example. So cell.maboss.myModel.network.symbol table, and then the name of the variable here could be DAVG. So to edit code snippets give you all these commands for you. Finally, we do time step to step MaBoSS. So here in my step function, I am doing self.timestep underscore MaBoSS, and this integrates the network by one step. That goes in the step function. So in my step function, for the purpose of this delta notch demonstration, how different is it from the antimony example? So here we have time step MaBoSS, and we also have a time step antimony equivalent. And here it's just a change in syntax, like nothing else will really change. Here where we do the loop over neighbors, we have the average of delta being the cell, which in this case is neighbor. That's the reference to our cell, dot maboss dot delta notch, which is the name of the network, and then the name of the node, dot state to get to the state of the node, and then we are summing this to our delta average variable. And then after we have our average, we pass this back to our network. So we do cell.maboss.delta-notch.network.symbol table, DAVG equals DAVG. So now we are informing our network. We are passing the value from outside the CompuCell3D to the inside the MaBoSS network. So to get the state of a node, dot state. Finally, stepping MaBoSS, self.timestep MaBoSS, and you do that every step. Hmm. This slide could go after the exercise, but okay, let's talk about this. What are the modeling opportunities with MaBoSS and CompuCell3D? So, pathways that involve cytokines, also pathways that involve uh, tumorigenesis, which is one of the examples I gave. And also a pro tip, there is surprisingly few papers, or there are surprisingly few papers using MaBoSS to building serious biological models. It was very hard to find one with the help of ChatGPT. And there are no papers on MaBoSS and CC3D for biological modeling. So probably our paper with APL from Johns Hopkins will be the first one. And I even asked an AI tool called Owl, which explores like millions of papers in the literature. And you ask the question, has anyone built a relevant biological model that incorporates both MaBoSS and Cellular model as core modeling tools? And the answer is no. It is very hard to find this AI to give you a no as an answer. So here, I'm pretty confident that nobody has ever built a serious MaBoSS plus CompuCell3D model. So you can be one of the pioneers if you publish a paper on a biological model that incorporates both MaBoSS and CompuCell3D. Okay, now let's get to our exercise. So our exercise is basically switching antimony for MaBoSS, and these are all the instructions you need to do. So first, you need to add an import random at the top of your simulation. Then you need to add your delta notch simulation and your delta notch configuration. So the network structure goes here, and then the configuration goes here. Then you have to substitute those syntax. For example, here we have add MaBoSS to cell instead of add antimony to cell. This goes in your start function after the for loop over cells. Then you have to modify the way you do your delta average because it's using antimony syntax. So here you have to do neighbor.maboss.delta notch, the name of the state, the node, and then dot state to get the state. And then pass the average to back inside the network by doing cell.maboss.delta notch.network.symbol table. The name of the variable equals the average of delta in the neighboring cells. And then, of course, we are using tracking fields. So remember, we are using this tool called tracking fields to keep track of the expression of delta and notch on each cell. So also, it's also different here. In Tellurium, we were doing something else. Here we have to do cell.maboss.delta notch and then the name of the node dot state. We pass that to our cell dictionary and tracking fields happens automatically. So this is all you need to do. I will give you 5 to 10 minutes to accomplish this task. And if you're having trouble, following this, feel free to just download the Delta Notch in MaBoSS simulation ready to go. It is on the student materials, and I will also paste it on the chat. So if you want to practice implementing MaBoSS networks, feel free to follow the exercise. If not, you can just download the simulation and go to the exercise. So here, um, simulations. I said symbol table to update the field. If I said that, I was wrong. To update the field, you do cell.mabos dot the name of the network, which is delta notch. Then you pass the name of the node dot state to get the state of the node. What was the symbol table for? The symbol table, we use the symbol table to get to pass to the network the average of delta, because the average of delta is something we get from CompuCell. The MaBoSS network doesn't know about the neighbor cells. The delta notch network is its own entity. It runs elsewhere. It doesn't know anything about CompuCell. So we have to pass information that we acquire from our agent-based spatial model into the network somehow. So this is what we're doing. We have a variable called delta average that we measure in CompuCell, and then we have to pass this variable back to the network. Can you please explain time tick again? Time tick is how often the MaBoSS will read the values of the nodes. So if you're stepping every Monte Carlo step and your time tick is 2, you're missing one step. And if you set time tick lower than 1, you're sampling the network more often. It won't give you much, I believe. But again, I'm not an expert on MaBoSS. So and I'm not 100% confident of time step and time tick. This is how I use them. I set both of them to 1. This is what I do. They always 1 in my simulations. I know exactly what's happening when they both of them are 1. So the inverse of the rate is the average time, and I have run a simulation proving that. It's all okay. But yeah, I'm not sure if I talked about this, but the inverse of the rate is the typical time. So if you have, for example, rate equals 0.1 steps, so probability per step, then 1 over R is your typical time. So here your typical time will be 10 steps. So this is for you to of course on average, because things are probabilistic. So let's say you are setting like on average my cell takes 10 steps to change state and die. It doesn't mean that all your cells will do 10 simulation steps and then die. They can die at the first step because it's probabilistic. But on average, it takes 10 steps. On average. So Clara is a mathematician. She can demonstrate that to us. So you, let's see, Anastasia has a question. So we can't just do something like this, cell.mabos dot delta notch delta. No, no, you cannot. Because you are not changing the node. You are changing, you're not changing the node. You are changing a logic. The logic acts on the node notch. So let me show you the network structure again, because maybe here's is where I'm losing you. Inside node notch, we are changing the condition of notch going up or down depending on the average of delta. If you take this value, the average, and you change delta, you have a completely different network structure. It's not delta notch anymore. You're basically saying cells that sense delta become delta, which is incorrect. And there is also another problem, Anastasia. DAVG, this average, is a float, so it can be like 0.4, right? And then you are saying that the node delta, which is a Boolean, is equal to 0.4? If you could assign the value by something like, oh, but what you said, that will be wrong too. Yeah, there are different syntaxes, so there's nothing I can do here. Yeah, they are different syntaxes. So this is a good question for TJ. This is how it was designed. Okay, so who has it running? I will launch the poll. I think I waited 10 minutes already. I waited much more than I should. So if you don't have it running, I encourage you to download the simulation ready or take your time and to implement it and uh because I need to follow up with the results. It looks like before, haha. Yeah, that's right. So Anastasia got it to work. Um, so next exercise, if you got it to work, is modify the threshold variable, D threshold, which regulates how susceptible the cells are um, at responding to extracellular delta signaling by expressing notch. So the idea here is that the lower the threshold, the more susceptible cells are to responding to delta signaling, and vice versa. So the higher the threshold, the less susceptible cells are to delta signaling. Which means that if they are more susceptible to delta signaling, there will be more notch cells. And if they are less susceptible to delta signaling, there will be less notch cells. And right in between, where the threshold, the activation threshold is 0.5, you have roughly the same numbers of delta and notch cells, which is pretty cool. So try to get that to work. This is our third exercise of today's lecture. And unfortunately, we will run over a little bit the break. I'll try to keep it down to 5 to 10 minutes, the other exercises. We do have a histogram plot exercise, which I will demonstrate, and I will go through the code to explain how to set up a histogram, but we won't have time to do a code along, unfortunately. So I encourage you to try the histogram plots as homework. And the interesting thing here with the histogram plots is that they make evident the differences between antimony and MaBoSS. Okay, so this is the exercise, and while people are doing the exercise of getting those different ratios of delta and notch cells, I will leave this slide up so people can catch up on the syntax. I see that people got it to work, which is fantastic. There are the different different ratios. So Johanna got low number of delta cells. Ronak got high number of notch cells, which is the same. There are the plots. Okay, we have the histograms there. All right. Okay. So now to do histograms in CC3D, this is how we do. So first, on CC3D Python, scientific plots a histogram. So this is again our to edit snippets. We have add histogram plot on start function. So this is to introduce the histogram plot in your simulation at all. So let me expand this. This is how it looks like. We create the window, and then here we add a plot. We can add multiple plots. In this case, I am adding one plot for delta and another plot for notch. Then we have to, yeah, so these are the plots. You can set colors, you can set transparency, um, yeah, and the plot name. The title of the plot, x-axis title, y-axis title. Then you need to give your histogram a list. So here the example is for pressure. So let's say you are tracking the pressure of a cell. You need to give your histogram a pressure list. So how do you populate this list? Well, you first initialize the list somewhere, and then you loop over all the cells and you append cell pressure to the list. In our case, we want to append the values of delta and the values of notch in each of the cells. In the step function, so same thing, we go to scientific plots, histograms, and then we add histogram step function. It will throw us a code that we put in the step function. And here is where we pass the list. So if you have a pressure list, you do value array equals pressure list. And you also set the number of beans to a certain value. You can change the number of beans on the fly as you populate more and more your histogram. So this is how it looks like. This is specific for cell pressure, so I'm not going to talk much about this. And you can also export your plots. So this works for histogram. It also works for the normal scatter plots. You right-click the plot and you select export. In export, you can export, for example, as a CSV file, and also you can export as an image. If you click copy, you get an image of the plot. And if you can select CSV. There is this extra option of selecting columns for editing your CSV file, which I'm discovering now. And then you click export, it will pop up a dialog box where you select the output folder for saving the CSV. Yeah. So for the exercise, unfortunately we won't have time to set the histograms ourselves. So I will go over the code here. I have so the two simulations that I put on the chat have histograms, both the Tellurium one and the MaBoSS one. So let me do open show project panel. And then here I have the delta notch simulation with antimony here. So here I have create plot win, create plots, two plots, one for delta, one for notch. Here I have color and opacity. Then I create a list of notch expression and a list of delta expression here in the start function. Of course, if every time you plot a histogram you want to forget the previous data, then you have to set them to empty every time you recalculate the expression lists. In this case, I want to keep track of the early expression levels, so I am defining them in the beginning. Then in the step function, I have to populate my two lists. So here I have a list for delta expression and another one for notch expression. And here I have my two histogram plots. Here again, I'm passing the list of delta expression and the list of notch expression. And I have exactly the same for my MaBoSS model. So here I have create window, create plot, initialize lists. Then in the step function, I have populate the lists and then pass the lists to the histogram. And I will run them. Uh, Let me run them side by side. So let me show you how to run something by side by side. So usually what I do is I just open two edits. So here I'm splitting screen, and on the right I have my, let me close this one. I have my, on the right I have my MaBoSS, and on the left I have my Tellurium. And so let me open this in player. This is my delta notch simulation. Let me remove this. Get out. Okay. Histogram of delta notch expression, and here the cell field. So uh, the initial values of delta and notch are 0 for some reason. Oh, okay. Now it's fine. Yeah, I randomly assign delta and notch values to the simulation. I guess that's what I do. I'm not sure. Let me see if that's what I do. Yes, I initialize a random. Oh, between 0.9 and 1. Oh, this is the Tellurium one. Okay, so let me run this one. Uh, Too fast. All right, finally I have them side by side. So let me get rid of these to get some space. Okay. All right. Let me show the delta expression, and I will run them side by side. And so, okay, so now what is the difference between the two? The difference between the two. Okay, it almost stabilized already, but so here we have it's so one simulation, the one in the right is MaBoSS, and the one in the left is the antimony one. I will stop them and run them again side by side. So starting with the antimony, what do you observe here? Ugh, what an ugly looking screen. Oh, forgot to select delta here. Okay, this is our histogram plots. And now on the right, our MaBoSS histogram plots. So what are the differences we observe here? Okay, Anastasia pointed out the one of the main differences, or maybe the main difference, which is MaBoSS is binary, while Tellurium has values in between 0 and 1. Kea answered, MaBoSS has an all or nothing way of modeling, so cells can only be in a delta or notch state. That's correct. Okay. What else do we observe? So let me give you questions here. Can I do? Okay, this is good enough. So why do the histograms look so different? Well, because MaBoSS is an all or nothing approach, so we have only binary states. This was already answered in the chat. Which one is incorrect? Answer in the chat. Which one is wrong? Okay. I like the answers in the chat. This is not a question we can answer. Okay? So yeah, probably neither or both. All models are wrong. Correct. Yes. So Anastasia changed views after Clara said both. All models are wrong. Good. How fast do they achieve equilibrium? Is there a difference on how fast they they achieve equilibrium? MaBoSS is faster. Perfect. Yeah, so these are all the questions, and feel free to follow up this exercise as a homework. I will be here to help you as always and answer the other questions as you see fit. I will, okay, yes. Another homework exercise is implementing cell cycle, cell growth and division. I have a summary of Boolean stochastic networks. So in the end, it's a high-speed and accessible version of ODE systems. It's a this then that kind of interaction, very simple. Half the number of parameters compared to antimony. All parameters are time rates, which is great. Nodes are either on or off, and it's much, much faster than ODE systems.
And Mark, Pedro, while we're waiting, Mark has one question, but time to equilibrium depends on rate up down, right?
Yes, yes, good catch. Yes, it depends on rate up and down. So this is a great thing to experiment. You can get your MaBoSS simulation, change the rates, the main rates, the ones that are not zero, the ones, because I have just two numbers, ones and zeros in the simulation, which means everything is very, very fast. If you change from 1 to like 0.001, which means about okay, 001 is 1000 Monte Carlo steps of typical equilibrium time. Your simulation will take a long time to equilibrate. Yes, so yes, it depends on the rate up and rate down rates. Yes.
Okay. Can you see on my screen?
Yes, can.
Okay. So, hi everyone. My name is Mark Brillo. I graduated this year from my master in biophysics, so maybe one of the youngest. And for my master project, I was supervised by François Ganer and François Fagotto. And for this hackathon, I'm proposing to extend that project to something more specific that I think could be interesting for the hackathon. So the project is embedded into embryogenesis and developmental biology. And here we were seeing the first stages in development of Xenopus laevis, where the first cell was dividing, and then we see gastrulation and neurulation, and this is around the first 24 hours of the development. If we cut the embryo and we look to the internal movement of tissues, what we see is that there are huge movements of different tissues. And basically, in gastrulation, it's crucial because we internalize the endoderm and mesoderm tissue. For my master project, what I did was basically doing a cell pods model of a part of gastrulation, going from stage 10 to stage 10.5, where there is already some tissue migration. However, there is this other paper by Jason Wen and Rudolf Bauer, where they studied one of the particular movement that appears in gastrulation, which is the vegetal rotation, which is the change in shape that we observe in the endoderm from one stage to another. And they observed that when they cut explants from the embryo, they still could see the vegetal rotation. And so it's not something that depends on the rest of tissues that are migrating, but happens itself. And so these cells are active, and this is something that I propose to model with cell pods and CompuCell. And so the modeling goals will be to start doing a bottom-up approach for this model and try to start with very few plugins. And so, of course, if we only have volume, surface, or contact constraints, we won't have any migration in a certain direction. And so we can explore different ways to do migration with links or extension potentials that we saw during the workshop. And also we know that in this paper is discussed that this is an amoeboid migration. So maybe we can use compartments or dynamic compartments as we saw the last days. And another point is that this is a 3D problem, although the video is 2D. And this is because there is not much migration between zeta planes, but it's something that we could explore. And something that I would be very interested is that we prepare some good analysis of these simulations on the pressure that there is in the cell, tensions, the eccentricity of the cells, velocity gradients. And this would be very useful because then we will be able to compare directly with the figures provided in the paper. And so I think it's a project that could be feasible to be done in CompuCell because I have already started to do it for my master project for 5 tissues, and here we will have few less. And the input will be precisely these 3 basic plugins, and from then everyone is able to propose how to explore this migration that we observe. And the output would be that we would understand the vegetal rotation without other adjacent tissue migration. And also we will have many code to do further analysis for any of your future simulations on cell properties. So that could be also recyclable in the future. And the idea is that by the end, we're able to report some of the characteristics that they found in this paper. And so I propose that we explore different migration strategies, different levels of cell description, and we prepare different plots to analyze the simulations. This is the main paper, but there are some others that you can take a look if you are more interested. And my background is I graduated in math and physics, and I did some modeling in molecular dynamics. But for the last 4 months, I've been in morphogenesis. And everyone is welcome. But if you are more a biologist, then it's a good experience for modeling. If you are good in image analysis, maybe you can help in building up the initial conditions for the simulation and also the plotting. And also, if you have a lot of experience with modeling, well, then I think it's a project that can combine several aspects of the workshop and could be also interesting to you. And the only disclaimer is that on Sunday I have limited connection and I'm in Europe. So there's this time difference to take into account. Thank you. If any of you have any question, happy to answer them.
Great, thank you. Any feedback from instructional team? Or any questions from anybody else in the room? Feel free to post them in the chat or unmute and ask. Pedro, what do you think?
Yeah. What if someone doesn't have any of those strengths on the slide? Are they still potential teammates?
Yeah, everyone. I mean, I
Okay.
tried to thought about any possible background. If I forget any possible background, please feel also included. It was not an excluding list.
Okay. Yeah, just a joke. So you're still deciding on the method of cell migration?
Yeah, a bit. Not really sure because also, so I think it's not difficult to reproduce the video with many plugins.
Mm-hmm.
We could put as many plugins as we want and it will be very precise.
Mm-hmm.
But ideally, I would like that everything is very well motivated from biology.
Yeah, because there is always this question of how do cells know where to go? And this is something that have to be very well grounded in the literature, right?
Exactly. So there is a lot of data about this in your experience. So the paper reports a lot of information. So how is the overall shape? I think I put that in the presentation. So for example, they are able to track how the shape is changing, which are the cells that are appearing from different planes, if there is cell division. They manage to describe the different velocity gradients that they observe in different cells. And they even go to the molecular level to describe this migration, and which are the components in the membrane that are crucial for this migration. And so I think that there is enough data to start trying that. And I have some experience already with the CompuCell and doing simulation with cell pods, so this might also help.